Alright, so today I'm just going to cover the basics of the immune system. Just want to forewarn everybody, this is not going to be a comprehensive review, but this is going to give you a lot of the basics between innate and adaptive immunity. So let's take a look at the differences. So here we've got our innate, and here we've got our adaptive. Alright, what's the difference? Well, we will know that our innate immunity is going to be innately there. It's always going to be there. So as soon as we get introduced a bacteria, a virus, a pathogen, our innate immunity will deal with it first. So our innate immunity is going to involve places like our skin. It's going to act as a physical barrier against the bacteria. We've also got mucosal sites, such as our lungs and the GI system. We're going to have mucosal sites, and that's kind of going to be the battleground of our innate immune system. Some of the players in that war are going to be macrophages, and then natural killer cells, NK cells, same thing. I'll get into these a little later. But for now, just realize that our macrophages are going to see something, they're going to phagocytose it, meaning they're going to eat it, and hopefully break it down. Our natural killer cells, like I said, we'll get into that later. So that's our innate immune system, immediately acting. Next we've got our adaptive. Our adaptive immune system, so let's say day 0 we introduce the bacteria. It may take 2, 3, 4, 5 days for our adaptive system to really kind of ramp up. So it's not going to immediately act. And we've got 2 arms to our adaptive immune system. You've got the humoral aspect, which is going to secrete antibodies. There are going to be 5 different subtypes. Or we've got our cell-mediated. Cell-mediated. So that's going to be the 2 different parts of the adaptive immune system. And we'll go into each of those later. But before we go into any of this, I just want to give you a background regarding MHC molecules. Hopefully clear up any confusion and hopefully clarify some things for future discussion points. So if you are watching this video for a test purposes, what I'm about to draw is a great test question. So CD4 positive cells, which we'll get to, are always going to match up with MHC2 molecule, and then CD8 positive cells will always match up with MHC1. Easy way to remember this, 8 times 1 is 8, 2 times 4 is 8. So remember, 8 is going to be your target. 1 and 8 are always going to match up. 2 and 4 are going to match up, always to get you that 8. But that's just a little memory aid. So what does all this mean? Well, let's take a look here. We've got stranded DNA. And let's take a look at that area right there. That is going to encode for HLA gene. There are different subtypes of the gene. You've got HLA-A, HLA-A, HLA-B, and C. All of those are going to create the same thing, MHC1. So I've referred to MHC a lot. What does that actually mean? Major histocompatibility complex 1. So that's going to be the first subtype. There's 2 different types, MHC1 made by HLA-A, B, and C genes. So MHC complex is just going to be a recognition molecule on the surface of the cell. So if we've got our cell here, I always denote my MHC just like that. No point to it, just keeps it clear in my mind. So this is going to be an MHC. So we've got our cell on its membrane. We've got an MHC complex molecule. And I'll explain the function of that later. For now, realize that all cells have MHC except for red blood cells. So pretty much every cell in the body, minus red blood cells, are going to express this MHC1 molecule. Likewise, we've got another DNA strand over here. We've got a different portion of the gene or of the DNA here. Let's say that encodes HLA, DQ, DP, and one other one DR. I guess I can write that in. So we've got 3 different subtypes up here, kind of like up here. Same thing. It's all going to create one molecule. That's going to create MHC2. All right, so what cells express MHC2? All cells do one. Only antigen-presenting cell, APC, antigen-presenting cells, only APCs express MHC2. So what is an antigen-presenting cell? Well, antigen, so it's going to find antigens or parts of fragments of things in the bloodstream. They're going to present them. So we've got our cell here. Antigen-presenting cells have lots of arms. What they're going to do is they're going to find a piece of antigen. It could be bacterial antigen. It could be host antigen. It's going to eat it. It's going to phagocytose it into the cell. It's going to break it down, and it's going to spit the fragments out onto its little arms and present it. That is an antigen-presenting cell. So I just want to be clear. That right there is going to be Mhc Complex 2 minus the red ball, because that's going to be the antigen. And then this is going to be MHC class 1. Okay, so what is the difference between 1 and 2? So let's focus this back onto our discussion about innate versus adaptive. So let's go to innate. Innate immune system, always there, always going to be there. We've got our macrophages. Like I said, those are going to phagocytose stuff. They'll be activated by a different molecule, which I'll get to later. And I'll do a summary slide with this written in again, just a little more complete. So for now, macrophages phagocytose extracellular bacteria and sometimes viruses. It's kind of a frontline runner. Also, we've got our NK cells. These I like to refer to as the Hall monitor. Why do I call it the Hall monitor? Well, I'll explain this. So, natural killer cells. It kind of has a tough sounding name. It's really not. All cells in the body produce MHC1. So, use your imagination. This is a cell in the throat. It is not a red blood cell, so it's going to express MHC class 1. Remember, this is my little annotation for MHC class 1. Here comes a little virus. This virus is going to come and infect this cell. So this cell has been infected. So we know that this cell has been infected with the virus. The virus has 2 different strategies that it can use. One is it can hope that it can be expressed. MHC1, oh, that's not the right marker. MHC1 complex, the cell is going to break down a little fragment of that virus, going to express it, and hope that it can survive this NK cell. Otherwise, what the virus does is, instead of being chopped up, some viruses can completely wipe out that MHC1 complex. So that cell is not going to have an MHC1 anymore. So we've got our natural killer cell come along. It's going to be the Hall monitor. It needs a Hall pass. So this cell is going to be expressing little fragments of itself. It's going to stick it up to the MHC complex, and it's going to present. Since it's itself, Hall monitor sees its Hall pass. It sees that it's natural body cell material. It's going to pass the test. It's going to move on to the next cell. Same cell, or same thing. The cell's going to have little fragments of its insides get expressed to the membrane. NK cell sees it. Hall pass is good. Finally, it's going to pass to this virus-infected cell. That virus has wiped out its MHC1 complexes. It does not have a Hall pass. So this natural killer cell now is going to secrete cytokines and other activating materials which are going to cause this cell to apoptose or lyse, and which causes that cell to die. Hopefully that virus won't be able to infect other cells. So the natural killer cells check MHC1 complexes to make sure that they're there, first off, then to make sure that they're expressing the right molecule. And if both of those factors are in, then it passes to the next cell. If it sees that there's an MHC1 missing, or if that MHC1 was presenting the wrong piece of information, such as a portion of that green virus, if it sees it's the wrong thing, that natural killer cell will come and lyse that cell. So this is part of the innate immune system. So remember, that's our MHC1 up there. Has nothing to do with the CD8 yet. Just remember that NK cells recognize MHC1 presenting cells. So now let's move on to a different subcategory, which we're going to make sure is. So we've got our adaptive immune system. We've got our humoral, and we've got our cell mediated. All right, so humoral we'll talk about in just a few more minutes. I'd like to touch over the basics of cell mediated right now. So right now I would like to go over some of the T cell lineage. So we've got our baby T cell over here, also known as a stem cell or a T cell precursor. Okay, so here's our T cell. It's going to go to the thymus. It's going to mature in the thymus. So our immature T cell is going to go to the thymus and mature. It's going to take one of two different possible routes. Still going to be a T cell. All cells off of this lineage will be T cells, technically. It's going to either pick up a CD4 positive or a CD8 positive marker. So this is kind of coming into our clinical picture up here. We've got our CD4 positive cells and our CD8 positive cells. This CD8 positive cell is going to leave the thymus, going to go to the body. It's going to be a cytotoxic T lymphocyte. And that's going to be a CD8 positive cytotoxic T lymphocyte. What's the importance of this? Well, cytotoxic T lymphocytes, remember our natural killer cells were the hall monitors. Our cytotoxic T lymphocytes are really going to be the Army or the Navy. They're going to be the big players in the immune system. If I was a pathogen, I'd be most worried about the cytotoxic T lymphocyte. And so if I was just any old cell in the body, and I'm with my MHC1, and I'm infected with an intracellular bacteria or a virus, this cytotoxic T lymphocyte is going to come along. The cell is going to break up fragments of everything that's inside it, whether it's normal cell stuff, virus cell stuff. Some of them are going to present that virus particle if they haven't wiped out the MHC1, because that was one of the strategies. This cytotoxic T cell with its CD8 is going to find its MHC1, and it will come and absolutely kill that cell. Cytotoxic T lymphocyte doesn't mess around. It's going to be your army cell. It's going to kill that cell, especially if it's presenting the wrong material. So we've got our cytotoxic T lymphocytes. That's going to be our CD8 positive cells. Our CD4 positive cells are going to be our other subtype of precursor T cell. It's going to leave the thymus as well, and it's going to form, again, two different cell types. Still going to be T cells. This one's going to be T helper 1, T sub H1, T sub H2. So you have a T helper cell type 1 and a T helper cell type 2. Alright, so what is the difference between these? I would like to note that T helper cell 1s are going to be used to activate macrophages. And then they're also going to be used to activate the cytotoxic T CTLs. So macrophages, like I alluded to earlier, they're going to phagocytose stuff. They're part of your innate immune system. They're going to always be around in the bloodstream and tissues. They're going to be looking for things to eat. Let's say it comes upon a really stubborn bacteria, refuses to be eaten, or it gets eaten but it won't get degraded. It's going to seek out the help of a T helper 1 cell, and it's going to present some of that material to the T helper 1. T helper 1 sees that distress. They're going to release a molecule back to the macrophage, so it's kind of like a, you show me what's going on, oh, you need help, I'm going to release stuff to activate you and make you stronger. So it's going to create a super macrophage, which should have more power to kill that pathogen. Same thing with the cytotoxic T lymphocyte. Even the army needs to call in reinforcements sometimes. It might call in a T helper 1 cell to release cytokines and other activating molecules to really activate that cytotoxic T cell and cause it to go into killing mode. So let's get rid of these arrows to help simplify things a little. That's T helper 1 cell. Our T helper 2 cell is going to be used to activate B cells. So B cells we haven't talked about yet. B cells are going to be involved in the humoral immunity. So let's go into that. Hopefully you have been following up to this point. This line, cell-mediated, involves this lineage, pretty much, the cytotoxic T lymphocytes. And then we're going to go into the humoral arm of the adaptive immune system. So those cytotoxic T lymphocytes, you don't want just these massive army hanging out in your blood all the time, because they can sometimes attack your host tissue. So they're not going to be activated until you have a virus or something that really ramps up your immune system on day 3 to 5. Same with your humoral system. You're not going to have antibodies that are just going to be continuously secreted. You are going to have continuously secreted antibodies, but you're not going to have just massive amounts at all times. So we've got a B cell lineage. Well, the last one was T. Now we're going to start with a B. So B stem cell, B precursor cell. All same thing. It's going to be an immature B cell. That is going to be found in the bone marrow. That's going to go, and it will go through a bunch of maturation steps. I don't think for the basic concepts that's important, but what's going to kick out is a mature B cell. And that mature B cell is going to be able to secrete antibodies. But it's not just going to secrete them at random. It needs to be activated first. So this comes in to our second aspect. We've got our antigen presenting cell. Remember, with all the arms, it's going to present antigens. It's going to see that extracellular bacteria in the bloodstream. And that extracellular bacteria is going to get phagocytosed. It's going to be chewed up, spit out on an arm, and all these different colors. So this MH2 arm is going to present that extracellular bacteria eaten up. We know that there's stuff in the bloodstream. And this is going to show kind of the fragments that it found in the bloodstream. So then we're going to bring in our T helper cell type 2, and that cell is going to come and it's going to see that this antigen presenting cell is presenting foreign material. That T helper cell is going to be smart. It's going to go find a B cell and that matches this antigen. And so specificity, it's not just going to secrete random antibodies, it's going to find the right mature B cell to secrete the right antibodies. That's going to kill this bacteria in general. This mature B cell is going to secrete immunoglobulins. And the immunoglobulins are going to be just another fancy word for antibodies. They're going to be the 5 different subtypes. The main ones are going to be A, G, and M. The others do come into play, but for the most part, IgM, IgG, those are going to be the big players. So this is going to be referred to as humoral adaptive immunity. Cell-mediated, you're going to have those cytotoxic T lymphocytes. Humoral, you're going to have the mature B cells secreting antibodies. So let's piece this all together really quick and hopefully get a good picture of what we just talked about. So you've got innate, you've got adaptive immunity. Innate, we've got our macrophages and our NK cells. Our macrophages are going to be wandering around in the bloodstream, finding stuff to eat. If it finds something that's really stubborn, our T helper, if I can find something better, our T helper 1 cells are going to help activate it, creating super macrophages. Remember, through the reciprocal, hey, I'm in help, okay, I'll help you mechanism. We've got our NK cells, which are going to be looking for MHC class 1 molecules that are going to be found on every cell. Every cell is going to present. If MHC1 is missing, or if it's the wrong thing, this NK cell, the Hall monitor, it doesn't have a Hall pass, it will die. Then we go on to our adaptive. In our adaptive, we have cytotoxic T lymphocytes. Cytotoxic T lymphocytes will be expressing CD8 positive marker on its membrane. That is going to be recognized by MHC1, again on every cell that is there except for red blood cells. It's going to present, it's infected with a virus. It's going to present a little viral fragment. Cytotoxic T lymphocyte says, hey, not in my part of the woods. Going to kill that cell. If it needs help, it'll also be activated by this Th1 molecule, sending signals to it to turn into a killer cell and ultimately kill that cell. Lastly, you've got B cells. Those are going to be your cells that secrete your antibody. How does it secrete the antibody? Well, it needs the help of Th2 cells. Those Th2 cells are going to be activated by antigen-presenting cells. So this is going to be an extracellular bacteria taken up into the cell, broken down into arms. So this way, these antibodies can be released into the bloodstream, specific antibodies, because we're going to match up the MHC2 to the Th2 as a specific reaction. This smart Th2 cell is going to go find a B cell that's specific to that fragment or to this this bacteria, secrete antibodies, so ABS antibodies, going to go find and secrete it in the bloodstream to kill that. So we have defense against intracellular stuff, mostly mediated by your cytotoxic T lymphocytes and natural killer cells. You've got extracellular debris, pathogens, antigens, stuff, and that's going to be really killed by your macrophages because they're out in your bloodstream trying to find these extracellular bacteria. Or later on, you can secrete some antibodies to kill that extracellular pathogen. Hope this helped. If you found this useful, please click like down below or subscribe up in the top left corner. Thank you very much.